we're going to go ahead and move on to Teng-Leong Chew.
Thank you, Janine. I would like to also echo Ron's and Kristen's gratitude for your effort in organizing this series of lectures. Can everybody see my slide okay?
Yep,
All
we
right.
see it.
So thank you very much, and a warm welcome to everybody. Today I want to tell you a story about an imaging center that has come to embody the very essence of Janelia's culture of science. The Advanced Imaging Center, or AIC in short, was created in 2014, truly as an experiment at the time. What makes the AIC unique is that it is an imaging center that houses only pre-commercial instruments developed at Janelia. And more importantly, it was built specifically for the outside world. So in short, this is an imaging center created not necessarily for Janelia scientists, but specifically for you. The question is, why would Janelia want to do such a thing, right? Who in their right mind would want to do something like that? So if you look at the life cycle of an instrument, so this is basically at the time when the instrument has just been fully developed, and this is when the paper is published. Now, if that instrument is very lucky and its potential is immediately recognized by a commercial company, the commercialization process in and of itself can take a very long time. And even when that instrument is now finally out in the market, most biologists, scientists would take another 1 or 2 years, and if you're very lucky at that, to obtain enough funding to actually buy that instrument before that microscope can actually make a scientific impact. And in the eye of Janelia and in the eye of the developer, that long delay is just unacceptable. And that is the reason why AIC was created, is to completely remove those intervening years so that instrument can be immediately be put in a position to make a scientific impact. And for that, we are extremely grateful for these two funding agencies, the HHMI, as well as the Gordon and Betty Moore Foundation in creating the AIC. So the AIC program, in a nutshell, is a really great opportunity for biologists to gain access to instruments that are not yet commercially or widely available outside. So to access the instrument, you have to write a proposal that will be peer-reviewed. And if your proposal is accepted, you come to Janelia, usually in 2 or 3 weeks period, and the cost of the experiment will be covered by the AIC within reason. And Janelia will cover the cost of your lodging. So in a way, we try to remove as many barriers as possible for scientists to gain access to these technology. So this is not a technology talk, so I'm just going to spend 2 or 3 slides going through some of the instruments that are available, and the details of that can be gained at the website. So we certainly offer super resolution microscopy. So the instruments that we have is an interferometry palm system. This is a 2nd generation super resolution microscope that was originally developed by Eric Betzik and Harold Haas. After they developed the 1st generation microscope, Harold continued to improve the axial resolution of the original palm system. And so these are the 2 movies in which details of biology is visualized by IPALM. So on the left panel, what you're looking at are 5 focal adhesion complexes. And what we're going to do now as we play through the movie, we're optically section through those 5 protein complexes at a step size of about 10 to 12 nanometers. So you're actually looking at the focal adhesion up and down. And as we tilt it to the side, you can see the red, which is paxillin, that are interacting at the extracellular matrix. So they are closer to the bottom, and then a signaling molecule that are located slightly higher up on the focal adhesion complex. On the right, you are looking at an image of a single bacterium. And here we stain the outer membrane red and then the inner membrane green. And as I tilt that bacterium to the side, you can see that IPALM is capable of resolving in all 3 dimensions in near isotropic resolution, both the inner and the outer membrane of the bacterium. And of course, we also have several light sheet microscopes. And if you look at the upper right corner, that is a video that is captured by using a simultaneous multi-view light sheet microscope developed in Philip Keller's lab. And this data is generated by Kate McDowell. And so what you're looking at in pink is endothelial cells. And when endothelial cells differentiated into the cardiomyocyte, we flux out one of the gene and they turn green. And if you focus on the right panel, you can actually start to see the folding of the heart muscle, and you can even start to see the chamber of the heart and before the heart starts to beat. And of course, if you are a cell biologist, you cannot really use a SimView light sheet microscope because the light sheet is probably too thick. It is really absolutely perfect for a larger sample, such as a mouse embryo. If you're a cell biologist, you need the lattice light sheet microscope. And so this is the video of the exquisite detail of the mitochondrial network in orange. And what you can also see, hopefully, is this a faint blue cloud of actin that is shuffling the mitochondria as if somebody is shuffling a deck of cards. We suspect that this is the mechanism through which the cells is trying to distribute the mitochondria number equally into the two dividing daughter cells. Now, you can also take the lattice light sheet and push it further deeper into the tissue if you equip it with an adaptive optics. And on the lower right corner, this is the latest instrument developed by Eric Betzig that is called Mosaic. And what you're looking at here is a lymphatic space that is located in between the ear and the brain of a zebrafish embryo. And this is with a system correction only before the adaptive optics kicks in. And clearly, this is when the adaptive optics clicks in. Now you can see the endothelial wall and some junk that macrophages like to eat, labeled in blue. Now you clearly see a macrophage moving around in the lymphatic space. They go through transendothelial migration as if the endothelial wall is not even there. You are now truly looking at immunosurveillance live in the context of the whole organism. Now, we also offer a correlative light EM microscopy technique. And the light microscopy part is a structural illumination microscope at cryotemperature. And then the EM part is the focused ion beam SEM. Both of them are developed by the Harold Haas group. And so you are looking at some neuronal epithelial cells here. So as we peel off this first layer, what you're going to see is the enhanced resolution provided by the structural illumination microscope. The green color is basically the cell-cell junction, and then pink is a cytoskeletal bundling protein. And then this orange color is the EM. So as we tilt to the plasma membrane, you can clearly see high and low density areas, electron dense area of the membrane. And so we pseudo-color them in pink and blue, and when we tilt it around, so these are structures that is probably not visible unless you have a system such as this. They're still trying to characterize to understand what actually gives rise to such a features and at the junction of two cells. So one of the things that I want to talk about is actually the service provided at the AIC. And one of the things that we are very proud of is the fact that we are able to maintain almost 85%, sometimes even 90% of experimental success rate, even though the projects we accept are usually selected based on their high risk, high gain profiles. And visitors come from all over the world with almost every specimen imaginable. That actually makes our life quite fun and exciting. I always say that AIC is successful because it is actually located at Janelia. Many visitors do not realize, initially at least, that when they're here, that they actually received the full programmatic support of Janelia. Even before you submit your proposal, we provide in-depth technical consultation so that we increase your chances of success. And before you arrive at Janelia, your animals will be shipped to Janelia, your cell will be shipped to Janelia. We'll get all those things up and ready for you so that when you arrive, you don't have to waste time getting your system up and running. And as you can imagine, this kind of operation, the failure rate can be high if you don't have a lot of stopgap measures. And so Janelia has thousands of cDNAs that are also conjugated with various fluorescent proteins, and even some of them are in model organism. We readily share this, and sometimes we very, very aggressively save some of the rescue some of the experiments. And when you are at Janelia, you are also have access to some of the newly developed probes. And Luke Levis, for example, several weeks ago gave a talk about tool building at Janelia. So a lot of those imaging probes are available to you. When you're at the AIC, the microscope time is completely available to you 24/7. We do not schedule two competing groups on the same microscopes. And even the application scientist and the data scientist times are dedicated to your project. Even after you have long left the AIC, the imaging processing and analysis expertise continue to support your project. And this map basically shows you the origins of the proposals that we have received. So we clearly have received a proposal from every continent except Antarctica. That remains my target. But one of the things that you also notice is that the proposal origins concentrate mainly on the first world or the developed world country. One of the continents that really, really stand out is Africa, which has 16% of the world's population and only one proposal coming from that continent. So that immediately backs the questions is whether or not open science initiatives such as this is only meant for the developed world. And we really don't think so. And we consider ourselves an imaging center that has a global user base, and we therefore consider ourselves having a global mission and responsibility to really, really push the imaging technology and science in general to every corner of the world. So how are we going to deal with that? So one of the initiatives that we created is this workshop called Imaging Africa. We ran this workshop earlier this year before the pandemic hit. So we basically, this is an image or 1 week long imaging workshop that is open to the entire continent of Africa. And any scientists working in a nonprofit organization in Africa are eligible to apply. We have 24 openings, obviously, because this is a very hands-on workshop. And to our great excitement, and probably causes some serious heartburn and panic, is even though we only have 24 openings, we actually have 711 applicants coming from 31 countries. So we actually have more countries represented than we actually have openings at the workshop. At the end, after a lot of filtering and screening, we accepted 24 students from 18 different countries in Africa. And so we run that workshop. And another interesting thing, while we were in Cape Town, we realized that there is a large swath of the population that live in township areas like this. This is a very sad aftermath of the oppressive regime of apartheid. And so there are a lot of a huge population living in quarters like this that have limited upward mobility. And so again, several weeks ago, you have heard a wonderful talk by Anastasia Koch from University of Cape Town. She leads this youth group called AWAZA that goes into this neighborhood and identifies students or young students with very high potential and try to give them as many opportunity and exposure as possible. So we thought, why not we do an outreach within an outreach while we were in Cape Town. So we actually brought some of these students in so that they get to spend some time with the AIC team member and the international teaching faculty, and also some of the African scientists. And we collaborate with Manu Prakash in Stanford and brought in quite a few of this faux scope, as you can see here. So this is pretty much a microscope in origami form, so the students have a blast trying to assemble this microscope together. And so, this is a young kid peering through a microscope, probably for the first time. And they get to bring this microscope home and share the excitement of science with their peers. And we thought that was a pretty cool idea. Now, we don't only do outreach in far corners of the world. We also think that it is equally important to increase microscopy accessibility and also strengthen the imaging community closer to home. Many of you are aware of the Eurobioimaging and global bioimaging and a lot of other countries and continents that have organized their entire imaging community. And people always say that, no, it is not possible to organize this vast imaging community in the United States because the community is just too scattered and too big. So we decided to run a conference at Janelia led by the AIC. And to our great delight, in 4 days, we not only managed to organize the imaging community in the United States, we actually extend that into Canada and down to Mexico. We formed this Bioimaging North America Society that brings together not only core facility directors, but imaging and data science expert, as well as instrument developer. And as of last evening, the Bioimaging North America is now a fully funded entity. Unfortunately, I cannot tell you which foundation funded it because we're waiting for their official press release. I can tell you that it's a young and very well-known charity foundation located in the Bay Area. And in addition to that, AIC also partnered with George Langford at Syracuse on this PAIR-UP program, and PAIR-UP stands for Partnering to Advance Imaging Research for Underrepresented Minority Scientists program. And so we are trying to reach out to a lot of the underrepresented scientists. And in fact, these two programs that is on your screen right now work perfectly together. We're mobilizing a lot of the imaging scientists from Bioimaging North America to get involved in PAIR-UP. And so I hope this offers a glimpse into how the AIC has encapsulated the very idea of open science that has been championed by Janelia for a long time. Not only do we open our door and welcome scientists from all over the world, but open science to us means bringing science and technology to whoever needs it, even in remote areas of the world. In short, open science is for everyone, not just for the privileged few. And that will remain the guiding principle of the AIC as we move forward to tackle even bigger plans in the near future. So stay tuned for our next exciting news. Thank you very much.
Thank you, Leong. That was a great talk and some really impressive programs. I want to start with a question about the proposals. Can you elaborate just a little bit on what sort of specifically you're looking for in successful proposals and how they're evaluated?
Yes, the proposal is actually extremely easy to write. It's about 1,000 words. So they are broken into several sections. So the most important one is the justification. Whether or not your experiment actually have the right justification to use the instrument. So any projects that we think that widely available commercial instruments can tackle, that weakens the justification quite a bit. And obviously, the other very important thing is whether or not our instrument can deliver the required results. The next important topic is actually the significance and the impact of the science. We are very agnostic when it comes to the theme of science that comes to AIC. So anything that have high impact in the field, we will consider that competitive. We are also specifically looking to push advanced microscopy into what we will call the underserved discipline. So areas of research that conventionally do not leverage the power of microscopy to address their questions. And last but not least, one very important factor is the quantitative approaches. While our microscopes are very good at generating eye candies, as you can see, generating just pretty images is not something that, this program is designed to do. So we want to make sure that you can get informative results, hopefully also quantitative results out of your data. But the best way to approach that is, frankly, just to send us an email. We will pretty much take over from there. We will contact you within 24 hours. We will sometimes have a 1 hour long in-depth consultation with you and coach you through the entire process.
Thank you. A question from audience member Joe McIntyre. Joe says, what a wonderful program. And she wants to know, as an open access facility, is any of the imaging data also open access?
Currently, at some point, once it is published, it is open access. Now, the focus ion beam SEM data is something that we're looking closely into to share with the community, because as you can imagine, EM data is less context or target specific, right? It is context rich, but target specific, less target specific. So it is easily can be mined by the entire community on various front. And so we're looking into sharing those data. We may give the scientists who initiated the proposal a head start, maybe 6 months to even a year and a half of head start. And after that, we will open up those data for public mining at some point.
Sounds reasonable. So the next question is, do you or your team maintain any ongoing collaborations that, initiated from a visit or a visitor to the AIC? Do you follow up with them, follow publications or success of the project?
Yes, I think that questions can be answered from several fronts. Let me talk a little bit more about the duration of the visit. Some people sort of misinterpret quite often, actually, that a 2 or 3 weeks short visit they interpret that as a single visit. That is not entirely true. We fully recognize that some of the biological stories takes more than one visit to complete. But we also recognize that you can generate sometimes 60, 70, 80 terabytes of data in 2 or 3 weeks at AIC. So after the first visit, some people write follow-up proposal, which usually have a very high success rate. And they come back and then they wrap up the project. We also think that that 4 or 5 months to 6 months of a buffer is a good time for people to actually digest their data before they come back with another proposal. We also have quite a bit of collaboration, especially on the data analysis front. As you can imagine, there are a lot of information that are discovery that sometimes even the PI didn't expect to get. And so we're going into uncharted territory that really required new algorithm or new method to analyze those data. And so a lot of those image analysis remain ongoing collaboration. Absolutely.
So Liang, do you have an idea, I'm sure you do, of how many visitors you've had to the AIC since 2014?
We have about 55 to 65 groups per year. So this is 6 years. So you can do the math there.
Yeah.
But not all of them are new groups, because like I said, there are also recurring visits as well.
Right.
So, yeah.
So with 60 groups a year, then you guys might have different groups visiting simultaneously, but using different equipment then.
Absolutely. Yes.
Yeah. That's great. Thanks. I think at this point we are going to end today's seminar, but I want to once again thank you, Liang, and of course, Kristen. The talks today were fantastic. We really enjoyed them. I also want to thank the audience for joining us. If you did not get your questions answered for either Liang or Kristen, you can head over to our Slack channel. The link is in the chat box. And they have promised me that they will check in over there to answer any questions that you have. Last but not least, I want to encourage everyone to join us next Wednesday when Life Science Across the Globe heads to Cold Spring Harbor Laboratory. And we will hear from Bo Lee and Richard Sever. So join us next week and we'll see you then. Thank you.